Hello everyone, my name is Sandra Timme, and in my talk I want to present a study where we investigate the complex interplay between two bacterial pathogens in co-infections. One of these pathogens is the well-known bacterium Staphylococcus aureus, which is a round-shaped gram-positive bacterium that resides in the human microbiota as a commensal. It can be community-acquired or hospital-acquired and can cause skin and respiratory infections. The second pathogen is called Acinetobacter baumannii, and it is a nearly round, slightly rod-shaped gram-negative bacterium that is exclusively hospital-acquired, and its natural habitat is still unknown. And it can cause a variety of different infections like pneumonia, bloodstream infections, or wound and surgical site infections. Both of these pathogens affect primarily immunocompromised patients, and both of them are members of the so-called ECCB group, which is a group of pathogens with high rates of antimicrobial resistance and that is responsible for the majority of nosocomial infections. When both of these pathogens occur in co-infections, this is associated with a worsened prognosis for the patient and increased mortality rates. And for these reasons, we are interested in elucidating the interaction mechanisms between those two pathogens during the co-infection. And here we have a special focus on the one hand, on the interaction via secreted compounds that happens in a contact-independent manner. Then, of course, we want to elucidate the contact-dependent mechanisms due to physical interactions of both pathogens. And thirdly, we are also interested in the interaction with host cells, like for example host cell invasion or host cell death. Due to time limitations, I want to focus on the first 2 points in my talk. In order to tackle these questions, our experimental collaborators performed a variety of different experiments, and first of all, they performed some control experiments where both of the two pathogens are grown in TSB medium, which is tryptic soy broth, and this is a high nutrition medium that provides optimal growth conditions for both of these pathogens. Secondly, they performed the so-called CFCM experiments, where CFCM stands for cell-free conditioned medium, and in order to obtain this, both pathogens were separately grown in an overnight culture, and then the cells were removed to procure the CFCM, which was afterwards used again as a medium to grow both pathogens in. So each pathogen was grown in their own CFCM, which we would expect would lead to the lowest growth since all nutrients are already consumed. And then also both pathogens were grown in the supernatant or CFCM of the other strain in order to investigate these contact-independent interaction mechanisms. Thirdly, our collaborators performed such co-cultivation experiments where both strains are together grown in TSB medium. From all of these experiments, different measurements were obtained, like for example, growth curves were obtained by measuring the optical density in these 96 well plates. And secondly, also the colony forming units were counted after 24 hours of the cultivation of the bacteria. So, and in this study, we did not want to restrict ourselves to using only one strain for each species, but we wanted to have a more comprehensive picture on their interaction, and that is why we chose for each species 2 different strains with a lower or higher virulence in order to investigate the general interaction principles. And furthermore, we also isolated pairs of Staphylococcus aureus and Acinetobacter baumannii from hospital patients where they occurred in the same wound, and these strains we used then also for these experiments I explained earlier in order to exploit the biological variation that can be found in the clinical setting. So, but first I want to show the results using these laboratory strains. And first of all, I want to show the results for the colony forming units. And in order to compare these different growth experiments, we decided to look at the relative change of, for example, the growth in the CFCM medium versus the growth in the control experiment with the TSB medium. And so for here a small example in order to show you how the results were obtained. So we have the CFU raw data for example, for A118 in the supernatant or the CFCM of the LS1 strain of S. aureus or in co-cultivation. And then we have the control measures with the TSB medium. So we can calculate the relative change, which you can see here. And from this, we can also plot these nice heat maps where the color refers to the median value of these distributions. And we can see how the growth was either reduced or enhanced in comparison to the reference experiment. So let's have a look at the results. So here in the first row you see the growth of the two pathogens in CFC medium compared to growth in the TSB control experiment. And what we see is that we have a strong reduction for both species here. When we compare the co-cultivation experiment to the TSB control experiment, we see that for S. aureus we have a similarly strong reduction, but for A. baumannii we have a much less reduction, so it's only slightly affected due to the co-cultivation of the pathogens. And thirdly, we also directly compared the measurements from the co-cultivation experiment to the CFCM experiments, and what we see is that A. baumannii has a huge growth advantage in the co-cultivation compared to the CFCM experiment, while S. aureus is still slightly impaired by this co-cultivation and the physical contact. So what we can say is that we see a competition via secreted compounds or the use of the same nutrients in the CFCM experiments. We also see that A. baumannii benefits from the physical contacting, while S. aureus is slightly impaired by the physical contacting. So next, we want to also have a look at the growth curves that have been obtained during the experiments. And here you can see in the first row the growth curves of the control experiments in the TSB medium. When we have a look at the growth curves of these species in their own CFCM, we see that there is nearly no growth, which is probably because all nutrients have been consumed. When these species are grown in the supernatant of the other species, we see that there is still growth to a certain extent, but less than in the TSB control experiment. And finally, we can also have a look at the growth curves in the co-cultivation. We see that we have equal or higher growth even than in the TSB medium, but however, here we cannot distinguish both species separately, but this is just the overall growth that we can measure. And now our aim was to develop a model in order to fit these growth curves and to quantitatively compare the growth rates of these species, and also to predict the subpopulations of both species in these co-cultivation experiments. And for this we set up an extended logistic growth model, because as you have seen before, sometimes these growth curves have this behavior of going down after some time of the experiment, and this we would not be able to capture with a normal logistic growth model. And in principle, our growth model has the same form as the normal logistic growth model, but what is different is that we have a time-dependent carrying capacity that varies between a start value and an end value, and it can decrease or increase over time with a turning angle at the time point P, and the gradient or the steepness at that time point is given by the parameter G. The motivation of this time-dependent growth rate is that we assume that the two species interact via secreted compounds, like for example certain toxins, or that nutrients are consumed, which changes the capability of the medium to hold a certain capacity of both pathogens. Then, of course, so for the CFCM experiments, we have then these 5 parameters. But of course, for the co-cultivation experiments, we simulate both subpopulations with these extended logistic growth model, and so we end up with 10 different parameters. The parameter estimation was done using simulated annealing with multiple starting points, and the fitted growth curves you can see here. So these are the simulated growth curves, and you can see that they fit very well with the experimental data. And now we can also make predictions on the growth of the 2 subpopulations in the co-cultivation experiments. So now we can have a look at the growth rates that we estimated by fitting the model to the experimental data curves. I want to only focus on these 3 values, which is the growth rate of the pathogens, the end value for the carrying capacity, and this additional value that we calculated, which we call C difference, which is the difference between the end and the start value, and which tells us whether the carrying capacity in or decreases over time. So when we have first a look at the growth in their own CFCM, which is corresponding to this main diagonal here in this subplot, we see that we have lowest growth rates and also the lowest values for the end carrying capacity. In the supernatant or the CFCM of the other strain, we see that for A. baumannii we have similar growth rates in the CFCM of S. aureus compared to the growth in the TSB medium. However, the end carrying capacity is much lower, and also we see that it is going down over time. On the contrary, for S. aureus, we see that we have much lower growth rates in comparison to the TSB medium. However, the end value for the carrying capacity is higher than for A. baumannii, and we don't observe any in or decrease, so the carrying capacity remains constant for S. aureus. And furthermore, in the co-cultivation experiment, we see very high growth rates for both species. And what we also observe is that the end value for A. baumannii is now much higher compared to the growth in the CFCM medium, while the opposite is the case for S. aureus, where we have lower end values than in the CFCM medium. And we also observe a different trend in the in or decrease of the carrying capacity. So now we see an increase for A. baumannii and a slight decrease for S. aureus. So with this we can conclude that A. baumannii might be impaired by secreted compounds in the CFCM of S. aureus, and S. aureus could be impaired by the physical contacting with A. baumannii. So further experiments in that direction could then reveal the underlying mechanisms, and for example, we could show that S. aureus produces quorum sensing molecules, like for example these phenol-soluble modulins that impair the growth of Acinetobacter baumannii. We could also identify that Acinetobacter baumannii might be able to also produce some toxins that interfere with the quorum sensing system of S. aureus. Furthermore, we also found a positive interaction between those 2 pathogens. So S. aureus is using glucose and as a byproduct produces acetoin, which can be cross-fed due to Acinetobacter baumannii as an alternative carbon source. Furthermore, we identified the mechanism during the physical contacting, and here Acinetobacter baumannii was shown to express the type 6 secretion system, which is able to, due to physical interaction, penetrate the other pathogen, in this case S. aureus, and directly transfer some effector molecules, like for example some toxins, to the other cells, and thereby impairs the growth of S. aureus. So next I want to present the results that we obtained for the patient samples, and here I first also want to show the results for the CFU counts. So on the left-hand side, you see again the results for the laboratory strains, and on the right-hand side, the results for the patient samples. So what you see for the growth in the CFCM, we have a fairly conserved interaction behavior. So most of these patient samples behave quite similar to the growth of the laboratory strains. On the contrary, when we look at the results for the co-cultivation experiments, we see that only one of the patient samples is in line with the laboratory strains, and this is sample 5. All of the other samples behave differently than the laboratory strains. So either as aureus seems to be much less affected, as you can see here, and baumannii seems to be more affected by the physical contacting in the co-cultivation experiments with these patient samples. And next we can also look at the growth curves for these experiments. So we see that the growth in the CFCM of the other species is very similar to the growth of the laboratory strains, while we have a completely different behavior for most of the samples in the co-cultivation. So now as aureus outgrows the Acinetobacter baumannii in most samples except sample 5, as we have seen before. So, and also when we look at the growth rates in comparison to the growth rates of the laboratory strains, which are shown in the upper row again, we see that both species have similar growth rates in the CFCM of the other species, while when we look at the carrying capacity related parameters, we see a complete switch of the behavior except for sample 5 again. So, and with this we can conclude that with our model and with the experiments together, we were able to identify the complex interaction mechanisms between S. aureus and baumannii. So we found several mechanisms that are acting on a contact-independent manner, like quorum sensing or cross-feeding of alternative carbon sources. But there are also mechanisms explaining the contact-dependent growth patterns that we have seen. For the patient samples, we could see that the interaction via the secreted compounds is fairly conserved, while the interaction via the physical contacting is for most samples very different to the laboratory strains, and we hypothesized that this could be due to a missing or non-functional type 6 secretion system in Acinetobacter baumannii. And with this, I would like to thank you for your attention. Especially, I would like to thank our collaboration partners from the Jena University Hospital. And I would, of course, also like to thank my research group, Applied Systems Biology at the Leibniz HKI in Jena, Germany. So thank you for your attention.